In this tutorial, we're going to look at protein synthesis. First, you're going to explain how genes carry information to make a protein, then describe the stages involved in transcription and translation, the two key stages in protein synthesis, and then explain how a genetic mutation can affect a protein. So proteins are probably the most interesting and useful family of molecules that exist in living things. Essentially, proteins determine the physical characteristics that an organism has. It may interest you to know that these three pictures here have something in common. The protein that makes the scales in this snake, or the horns of this rhino, or the hair on this girl are exactly the same. It's a protein called keratin. In fact, in many hair shampoos on the front it will say it contains keratin, sort of liquid hair if you like, for repair of damaged hair. The horns of a rhino are in fact fused hair filaments. And hair or fur is just evolved scales. Our general understanding of proteins has been misled or misdirected by dietitians. People normally think of muscle when we think of protein, but the proteins that make muscle are only one example of structural proteins. Proteins are far more interesting than that. You see, if you removed the water content from your body, 90% of your dry mass which is left would be protein. More commonly, proteins can be enzymes, so most proteins in your body are enzymes. Enzymes are proteins found in living things which speed up chemical reactions to a rate which makes life possible. Without enzymes, chemical reactions in our body that keep us alive, such as respiration, would happen so slowly that we would die. Many poisons that kill you actually affect your enzymes directly, like cyanide. Proteins can also be hormones, chemical messengers. If you remember from the lesson on glucoregulation, insulin and glucagon are examples of such hormones. And proteins can be structural, so they can form structures such as your fingernails and hair, which are made from the protein keratin. So proteins are made from amino acids, monomers, tiny molecules called amino acids, which bond together to make a polypeptide or a long chain of amino acids we call a protein. There are 20 different types of amino acids found in the body. The number and sequence, in other words the order of these amino acids, determine the type of protein that is made. This is one of the most important facts which you need to understand the process of protein synthesis. So these shapes represent some of the 20 amino acids found in our body. Some of these amino acids we can actually make within our body, but some we must acquire from the food we eat. These proteins are called essential amino acids because we can't make them and we need them to survive. So here I have three examples of protein. Keratin, the structural protein that makes hair, insulin, the hormone that regulates blood glucose levels, and the enzyme amylase, which breaks down starch into glucose, or rather assists in the breakdown of starch to glucose. So let's say these amino acids, when bonded together, will make the protein keratin. And let's say when we stitch or bond these amino acids together, we make the hormone insulin. And finally, if we stitch these or bond these amino acids together, then we make the protein amylase. So you can see what sets these proteins apart is 1, the types of amino acids that make them up, and 2, the order in which they appear. Some proteins may have more amino acids than others. The sequence of amino acids determines how this protein chain will fold to make a three-dimensional structure. If you change the sequence of amino acids, you will change the three-dimensional structure of the protein and therefore affect its function. So, how exactly are genes involved in making proteins? Well, firstly, remember what a gene is. Genes are sections of DNA that determine the sequence of amino acids in a protein. You could also say genes are sections of DNA that code for a specific protein. The message in a gene is understood 3 bases at a time. These 3 bases are called triplets or codons. So here you can see a section of a DNA molecule we call a gene. And the reason why this is a gene is because it codes for a specific protein. Here are the bases that make up the gene, the 4-letter code that makes up DNA. Adenine, cytosine, guanine, and thymine, and you can see the sequence here. Now I've put these imaginary lines here just to divide up the triplets. So here we can see one DNA codon, another DNA codon, and another one. In exams, they can sometimes give you a section of DNA and ask you how many codons there are, and you just need to count them in 3. So this would have 3 codons. Think of DNA like a cookbook, and think of each gene as a recipe in the cookbook. Now, you would never say a cookbook makes cakes, for example. Rather, you would say the cookbook contains the information to make the cake. But you still need a chef to make the cake, to turn that information into a physical thing. So the cell's chef is actually an organelle, a part of a cell called a ribosome. So DNA codes for proteins, or rather a gene codes for proteins, but the ribosome makes the protein. So the first part of protein synthesis is called transcription, and it occurs in the nucleus. So DNA is part of a family of molecules called nucleic acids. Just like proteins are a family of molecules, carbohydrates are a family of molecules, and fats are a family of molecules, nucleic acids are another very important family of molecules found in living things, and their specific job is to store information. So during the first part of protein synthesis, in other words transcription, the information on DNA is coded onto another nucleic acid called mRNA. mRNA stands for messenger RNA because it carries the message which DNA has. Similarly, I've divided the mRNA molecule into triplets called codons as well. Now, unlike in DNA where A, adenine, always pairs with T, thymine, in mRNA, wherever you'd normally have a T, you replace it with a U for the base uracil. Uracil is only found in RNA molecules, never in DNA molecules. Some exam questions require you to decipher the code on DNA to construct an mRNA molecule. So basically what we're doing is we're changing the language of one nucleic acid into a very similar language of another nucleic acid. It's largely the same with just a few key differences. So it's a bit like changing one language like English into American English, largely similar but with a few different words. For example, we say autumn but Americans would say the fall, or we say tap, the Americans say faucet. This is why we call it transcription. We're just changing it into a very similar but slightly different language. The second part of protein synthesis occurs in the cytoplasm and it's called translation. We need to find an organelle called the ribosome, which I described as the cell's chef, to carry out this stage. So what will happen is the mRNA will bond with the ribosome. Each mRNA codon will attract another type of nucleic acid called tRNA. tRNA stands for transfer RNA. But each tRNA molecule will be associated with a specific amino acid. When brought close together, the amino acids bond together to make a protein. This is why we call this stage translation, because we're changing the language of nucleic acids into a completely different language, the language of proteins. So it's a bit like taking our American English now and translating it into French, a completely different language with completely different words. So just to review, transcription is the first stage and it happens in the nucleus. Here, the message on DNA is encoded onto a molecule of mRNA. The mRNA then leaves the nucleus and joins with a ribosome, where it will attract specific molecules of tRNA with their associated amino acid. The amino acids then bond together to make a protein. I will go into all these stages in more detail, but this is a really good overview for understanding the key parts of protein synthesis. So remember, the sequence of triplets in a gene determines the sequence of amino acids in a protein. This is the most important fact of the lesson. Also, be aware that genes can be switched on and off by our cells. This means that different cells produce specific proteins. For example, only pancreas cells produce insulin. The gene for insulin is found in every cell in our body, but it's switched off. The only place where it's switched on is in the pancreas. In the salivary glands, you'll find cells that produce the enzyme amylase. And in hair follicle cells, you'll find the gene switched on to produce keratin. So gene switching determines what the cell does. So in other words, gene switching makes cells specialized. I mean, it'd be incredibly wasteful if your pancreas cells, for example, produced keratin. There'd be no point in it. Why would you want hair growing out of your pancreas? So the body saves energy by switching off genes where they're not needed. So that is how we explain how a gene carries information to make a protein. Remember, it just carries the information. The ribosome makes the protein. So now let's look at the process of transcription in more detail. You see here we face a biological dilemma. DNA is the molecule that carries the information to make a protein, but the ribosome makes it. Now DNA is a huge molecule and it's very important to us. We don't want to risk it leaving the nucleus so it could get corrupted and destroyed. It's way too important. Also, it's so huge it physically can't get out of the nucleus. This is the nuclear envelope with the pores, and you can see DNA is huge, it can't leave. So, how do we get the information stored on DNA out of the nucleus to the ribosome so the ribosome can make the protein? Well, this is precisely why we need another molecule called mRNA, messenger RNA. It's much smaller and can easily leave. So the first stage of transcription involves the DNA unzipping. You can say that, DNA unzips. What that really means is the weak hydrogen bonds, which I've drawn with white dots here, you can see here weak hydrogen bonds, they break. It's very important that they're weak because they need to break and unzip DNA frequently. So let's see that. There you go, DNA has unzipped. Now, only the gene unzips. The rest of the DNA will still be coiled up within the nucleus. This gives the cell a bit of control over what proteins are made. The next stage involves an mRNA molecule being made from a DNA template. So this is our DNA template, and these are the single monomers that make up mRNA. So these are called nucleotides. And just like any component of biological molecules such as amino acids and glucose, we get them from our food. So mRNA nucleotides are free floating in the DNA and they'll be attracted to their complementary base on DNA. So uracil will be attracted to adenine, guanine will be attracted to cytosine, cytosine will be attracted to guanine, G to C here, until we've made our molecule of mRNA, messenger RNA. You can see how it's smaller than DNA. Its small size gives it the advantage of being able to leave the nucleus. So the next stage is mRNA leaves the nucleus and enters the cytoplasm. So you can see it's leaving through a pore in the nuclear envelope and it's in the cytoplasm now. So that's all you have to remember for transcription. DNA unzips, mRNA molecule made from a DNA template, and then mRNA enters the cytoplasm. You don't need to talk about individual nucleotides or anything like that. Stick to these 3 sentences. So as I said before, the next part of protein synthesis is called translation, and it occurs in the cytoplasm, and it requires a ribosome to make the protein. So the first step is mRNA joins with the ribosome, so it's locked in place. The ribosome is a bit like a workbench which clamps mRNA in place. Now it's time to introduce you to the other nucleic acid, tRNA, transfer RNA, but you can think of it a bit like taxi RNA because it shuttles amino acids to the ribosome. Now tRNA has a very different shape to mRNA. It has a cloverleaf type shape. All around it you'd find bases, but the only ones you're interested in are the ones at the bottom. Here you'll find 3 bases, another triplet, but this time we don't call them the codon, we call it the anticodon, because it will be complementary to the codon on mRNA. Each tRNA molecule with its specific base sequence will have a specific amino acid associated with it. So what happens next is mRNA codons, here they are, 3 of them, attract complementary tRNA anticodons. That's probably the most complex sentence in this whole procedure. So the first codon on mRNA is UGC, uracil, guanine and cytosine. So they need their complementary base pair, so it'll attract this tRNA molecule. So you can see A, adenine is complementary to uracil, cytosine is complementary to guanine, and guanine is obviously complementary to cytosine. These white dots, once again, represent the weak hydrogen bonds which temporarily join them together. And exactly the same thing happens to the next codon, which will attract its complementary anticodon, and the next one, which will also attract its complementary anticodon. Remember, each anticodon on a tRNA molecule is associated with a specific amino acid. When the amino acids are brought close together, in close proximity with each other, they form a different type of bond called peptide bonds. This is why a protein is also called a polypeptide, because it has many peptide bonds. So remember, each tRNA molecule carries a specific amino acid, and the amino acids bond together to make a protein or polypeptide. What will happen next is the tRNA molecules will unattach from the amino acids and go off to collect more of the same type of amino acid. This is why I think of them like taxis, because they shuttle amino acids to and from the ribosome. So remember, in transcription, DNA unzips, an mRNA molecule is made from a DNA template, and mRNA enters the cytoplasm. This happens in the nucleus. In translation, mRNA joins with the ribosome. mRNA codons attract their complementary tRNA anticodons. Each tRNA carries a specific amino acid. Amino acids bond together to make a protein or polypeptide. Obviously, this is a very simplistic example. Proteins actually contain many more amino acids. And that is how you describe the stages involved in transcription and translation. Finally, let's see how genetic mutations can affect this process. A genetic mutation is a change in the base sequence of DNA, and this can affect the sequence of amino acids in a protein. As I said before, the sequence of amino acids affects how the protein folds and bonds together to give it a specific three-dimensional structure, also known as its tertiary structure. And this affects the function the protein has. So here you can see different sequences of amino acids, and you can see how this results in the protein folding in a different way to give it a different three-dimensional shape. So a mutation can affect the sequence of amino acids in a protein and therefore affects how it folds and therefore performs. So for this example, I'm going to focus on the last amino acid sequence and this protein that it makes. So this is the normal base sequence for the gene, and here the amino acids each codon codes for. And this will result in an enzyme with this shape, or a protein, but I'm using an enzyme as an example, with this shape. Now enzymes act on specific molecules called substrates, and you can see that they have a complementary shape, so this substrate fits into this enzyme. Now if the shape of this enzyme was to change, maybe the substrate would no longer fit and the enzyme could no longer do its job. So firstly, let's look at the idea that mutations can be harmful. So here you can see the normal base sequence, but a mutation has caused a change. A mutation at this point has caused a change in the base sequence, so we no longer have thymine here, we have cytosine instead. This results in a different amino acid being coded for at this point. The other amino acids are the same, but this one is different due to this mutation. This affects the three-dimensional shape of the enzyme it codes for. So remember, this was the original shape, but now with the mutation, it folds like this. The point is, it no longer fits the substrate and therefore cannot do its job. So a real-world example of this could be, for example, the enzyme lactase, which breaks down the sugar in milk, and if it mutated, then it would no longer be able to do that, and you'd have a problem called lactose intolerance. But let's say if it was an enzyme involved in respiration and it could no longer bind to its specific substrate molecule, that could actually result in your death because respiration is essential to keep you alive. So now let's look at the idea that some mutations can be neutral. They have no overall effect. This can be interpreted in two ways. So once again, I've got my change in base sequence and mutation here. But believe it or not, sometimes the same amino acid can be coded for by different codons. So every amino acid has at least 3 to 4 codons that code for it. This is a great safety backup mechanism our body has. So the overall effect is the enzyme made has exactly the same structure because it has the same amino acid, so it folds in the same way. But I said it could be interpreted in two ways. The other way is the change is neither harmful nor beneficial. So for example, here we do have a different protein being coded for, but let's say we're looking at earlobes. Some people have attached earlobes, like this person attached to the side of their face, whereas other people have dangly earlobes with their earlobe not attached to their face. Now, correct me if I'm wrong, but I don't think this is a harmful or beneficial mutation. It certainly doesn't increase your chance of reproductive success. At least I hope not. And finally, some mutations can be beneficial, and this is the most exciting part of protein synthesis. This is what drives evolution forward. So once again, I've got my mutation, and this time it does code for a different amino acid, but the overall effect will be beneficial, as you will see. So let's say we're looking at antibiotic resistance gene in a bacteria, and let's say this was the enzyme that basically the bacteria initially had, but after the mutation, the shape has changed because it's folded differently. Now the bacteria has developed the ability to break down an antibiotic it couldn't before because they have a complementary shape. This is how antibiotic resistance evolves in bacteria. So it will increase the chance of the bacteria surviving and passing on this gene to the next generation of bacteria. So remember that mutations can be harmful by changing the shape of a protein so it no longer functions. They can be neutral by having no overall effect on the protein coded for, and they can be beneficial by changing the shape of a protein so it can now perform a function it couldn't before. And that is how you explain how a genetic mutation can affect a protein.